So I will give you an overview of the different applications that exist in the next-gen sequencing world. I will show you how you can prepare your library. Not everybody will have to do it himself. Sometimes your sequence provider will do it, but it is important that you have a notion on how it's done, because some of these things you need later on during the analysis. I will explain how it works, Illumina sequencing, and I will give a very brief overview of different sequencers. So first, relatively simple question, what can you sequence, or what do you sequence when you do next-gen sequencing? Yeah, so this was a very easy question because all the answers are right. Yeah, I saw that most of you go for whole genome sequencing. Indeed, you can do that, but that's quite expensive because you have to sequence a lot. So you have to make a lot of reads and this will cost you money. So you also have people who only sequence exons because that's much cheaper. It's only a fraction of the genome that you have to sequence then. There's also people who are more interested in transcripts. So that's what they sequence, the messenger RNA. And you also have people who are sequencing small RNAs. So all these things can be sequenced. Purely practically, what you sequence is a library, and that library will always consist of pieces of DNA. So whatever you start from, the thing that you sequence is a set of pieces, fragments of DNA. Each sample that you want to sequence will give rise to one library. So typically you have multiple libraries that you're going to sequence. So, and then depending what exactly you sequence, the application gets a different name. So when you sequence the whole genome, it's called whole genome sequencing. When you sequence specific regions in the genome, like regions that bind to a certain protein, it's called ChIP-seq. When you sequence specifically the exons, it's called exome sequencing. And the same is true when you start from RNA. So nobody directly sequences RNA because it's too unstable. So this is why also RNA is first transformed into DNA and then this copy DNA is sequenced. And also here, depending on what exactly you sequence, the application gets a different name. So when you sequence the transcripts, the messenger RNAs, it's called RNA sequencing. When you sequence the small RNAs, the microRNAs, it's called microRNA sequencing.